All right, hi everyone. All right, so it's just me tonight. So this will probably be a little shorter of a review session. We'll be going over the Spindle Assembly Checkpoint, the Anaphase, both of them A and B, and then also we'll be ending off on Cytokinesis slash uh, Telophase. And if any, it's just you guys here tonight. So whenever you guys have any questions, feel free to just unmute yourselves. All right, so quick overview of what we'll be going over again. It's going to be the Spindle Assembly Checkpoint, also known as the metaphase anaphase transition. We'll be looking at the roles of the different proteins and their functions within the checkpoint, what they do when they're active, when they're inactivated. Um, some examples that will be BUB, MAT, CDC20, and then MCAC. Then we'll be looking at anaphase. We'll be looking at Anaphase A, which is more focused on the chromosomes being pulled apart to the poles, and then Anaphase B, which is more focused on the poles being pushed and pulled towards the edges of the cell. And then we'll end off on cytokinesis, which basically will be going over the cleavage furrow and the levels of CDK and like what that changes for the cell and how that prepares it for the next round of G1 and the next cycle. So to start off on the spindle assembly checkpoint, basically the main function of it is just to ensure that all the sister chromatids are properly attached to their MTs, to their microtubules, right? Because that's really crucial for the cell because we don't want to have, say a case like non-disjuncture where one of the two daughter cells gets more DNA than the other, or just like any other like unequal sharing of like material, right? So basically the cell just wants to make sure that A, all the chromatids are lined up properly, and B, that they're all that they're all attached to the microtubules at the kinetical. And the way it does that is by sensing tension that comes from the tug of the microtubules being attached at the kinetical. So after it can sense that, then it knows that, okay, everything's set and ready and I can carry on to anaphase. Um, and pretty much the main proteins that do this sort of check will be your MAD and bulk proteins. You don't need to know the mechanism for how they go about it, but basically you just need to know that they'll be active during the checkpoint, and they're just going to be ensuring that everything's set up properly. And the way they do that, I just have a figure here from the lecture or from the videos. When they're in their active state during the checkpoint, they're going to sequester CDC20. So they're going to keep it out um, insoluble. So if you look here, this is going to be your kinetical, right? It's going to be made up of 80 proteins. And basically, after the cell can sense that, okay, so now all the chromosomes are feeling the tug or under the tension, then it's going to inactivate the BUB proteins. And what that will allow is for the CDC20 to be soluble, and then it'll be free to bind to APC complexes. And that'll basically start the transition towards anaphase. So basically what APC is going to do is that it's going to be marking, it's basically ubiquitin mediate choroialysis, right? So it's going to be starting to mark securin for degradation. And if you look here, you can see that securin, it's easy to, by the name, you can kind of use that to remember, right? Securin is going to be securing or inhibiting separase in place. And basically, when it's going to be degraded, right, separase is going to be free. It's going to go into the to the sister chromatids, and it's going to cut through the cohesin rings. So you can think of that separase cutting through the rings, and that basically that way the two chromatids will be free to separate from each other, and then the cell is going to be free to go into anaphase. Any questions so far? I feel like I went through that pretty quick. But if not, we go on to anaphase A B. So basically, anaphase is going to be divided into two stages. one focused on the chromosomes, and then B going to be focused on the actual spindle poles. And in different organisms, it can happen in a different sequential order, right? A can happen for B, or B can happen for A, or even both of them can occur simultaneously. If you want to look at anaphase A, this is going to be when the kinetic core microtubules are going to shorten, right? They're going to start walking towards that minus end, which is going to be at the ends here, at the spindle poles. But what also you're going to see is that if you go back to kinetic core, you're going to have MCAC, and what it's going to be doing is, since it's at the plus end, it's going to be taking off tubulin of the on the microtubule, so that's going to be like a controlled way for the cell to shorten the microtubules on that end as well. But it's got to be pretty well regulated, because you don't want MCAC to be taking off tubulin pieces, whereas then the microtubule can't even connect to the kinetic core. So basically, yeah, just those two things. Microtubules are going to be walking towards the minus end, and then over at the plus end, you're going to have tubulin being teared off by MCAC. And then anaphase B, basically you're going to have two things happening. You're going to have the spindle pole itself being pulled towards the edge of the cell, right? And then you can also have it being pushed by the interpolar microtubules. So basically on that side, you're going to have egg 5, and it's going to be sitting on the interpolar microtubule and going to be pushing it off that way. And then you're going to have dynein or some other minus end directed microtubule pulling the spindle pole towards that other side. And it's this like opposing forces of like push and pull that are gonna both work together to, again, like further separate chromosomes and the spindle poles. Then you're gonna have cytokinesis, and basically here it's gonna be a few things. You're gonna have the cleavage furrow, right? It's gonna be formed by the contractile ring, and that's gonna be what pinches off the cytoplasm, so that at the end you can have 2 daughter cells. And you can also have, since this is going to be basically the start of a whole new cell cycle, you're going to see things go back. So you're going to see spindles obviously break down, right? You don't need those anymore. And you're also going to see, this is a major like important part, you're going to see the decrease or decline of active CDKs. And this is really important, because CDKs are what really started the whole thing in the first place, right? They're the reasons that the chromosomes were condensed. They degraded the nuclear envelope, and they also have PRCs. So basically, by decreasing their activity, you're going to see the cell's going to kind of go back to that G1 phase, with the envelope forming chromosomes kind of decondense, and that way you can start the whole thing over with pre-RC's formation. But yeah, that's pretty much it on that video. Do you guys have any questions?
Not really. I think it was all covered pretty well.
Yeah. All right.
Oh, actually, wait. Now that I think about it, I actually do have one quick question.
Yeah. Go.
It's just a clarifying question. So for